This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 519, recorded on November 9th, 2018. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me here in New York City, Dickson Despommier.
Hello there, Vincent.
How have you been?
Well, thanks.
I just saw you 5 minutes ago. I don't know why I asked you how you've been.
Well, it's a minute-to-minute basis of life right now, I guess. Maybe, who knows? It's a weird world out there.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And it is, I hope I can say that the weather is really crummy here. It's
ours is
rainy and cloudy and 45 Fahrenheit and 7 C.
And we got the same crap here.
From Madison, New Jersey, Brianne Barker.
Hi, everyone. It's great to be here. And I will just say we have the same exact weather here as well.
Right. In fact, I can't see your house now.
No, you definitely can't see us.
Nope.
And from Austin, Texas, Rich Condit.
Howdy, everybody.
Hey there, Rich.
And we don't have the same weather here.
No.
But it is chilly. It's 56 degrees, which is Fahrenheit.
That's
13 C. Sunny, though.
Yeah.
There's a cold front coming through. Fall we are in fall's grasp here. It's actually, the forecast is for one of the lows next week to be 32 degrees.
Ooh.
There you go.
So there you go.
There you go.
Well, Kathy texted us just a while ago, said she had snow in Ann Arbor today.
Uh-huh. And I heard from one of my Buffalo friends, so they got snow in Buffalo.
Oh, yeah.
So here we go.
This episode is brought to you by the graduate and postdoctoral programs at the Department of Microbiology at the Icahn School of Medicine at Mount Sinai, composed of over 20 virology labs, all centralized in one building in the heart of New York City. The department is a perfect fit for anyone with an interest in pursuing virus research. The department is presently looking to recruit prospective graduate students to apply to the program by the December 1st deadline. That's less than a month.
It is less than a month. Interested
postdocs are also encouraged to contact faculty of interest. For more information about the department, go to bit.ly/micro-MSSM. That's all one word.
Are faculty of interest persons of interest?
Should be, right?
Yeah.
Depends on what they've done.
This is my old department. That's where I got my PhD.
I bet they have Wi-Fi there.
Stop it. Just stop it.
They might not because HIPAA regulations,
right.
Whenever there's a hospital, it has to be secure.
Yeah.
We have Wi-Fi on our floor, but it's too far away to get a signal in my office.
Right.
And it's a public Wi-Fi line, so it's limited in what you can do.
Well,
there is a secure line, but it never works.
Did you know that there's an entire
before
country?
the show, I was laughing hysterically about how typical this is of Columbia University to have cutting edge science going on in an environment that lacks what everybody else considers basic technology.
True.
Yeah, it has to be secure, so you can't steal.
But
people's
there is
health
such a
records.
thing as secure Wi-Fi. This is totally done in corporations around the world.
They have it. They have it, but
and hospitals.
I can't get onto it.
Yeah,
I tried, but the router is too far away. I would like to ask everyone to subscribe to my YouTube channel, youtube.com/PROFV as in Vincent RR, because I need 25,000 subscribers to apply for educational grants. You too, Alan, you need 25,000 subscribers.
I was going to say I subscribe to your channel. I don't think I'm going to get 25,000 subscribers on mine.
Someone told me that
since I've posted like 3 videos to it.
Someone told me that they misunderstood the V and typed something else in, and they got to a YouTube science channel with 200,000 subscribers by mistake, and he said, that's where all your subscribers are going.
Oh, no.
Oh, you're kidding.
Prof was in a short for professor, and VRR are my initials. Vincent R. Racaniello, Prof. VRR.
Should put a space there.
That's okay. It's okay. No problem. Just check it out. You might like it. It's free. Just subscribe. I'm not sure we're going to make it, but we can try. I also would like to point out that Susan Nassif has a virus comic book, which she released some time ago, called The Adventures of the Ragaches Volume 1 Vaccines. Please help support her work. I'm going to put a link to the show notes to her book, which is on sale for $9.95 US in the US. It's on Amazon, and it'll be in the show notes. Please help her out. Some time ago, I mentioned a clinical trial here enrolling at Columbia University Irving Medical Center to determine the effect of an anti-herpes simplex virus, antiviral valacyclovir. This inhibits the viral DNA polymerase on Alzheimer's disease. And if you've noticed, herpes simplex and Alzheimer's disease has been in the news lately, so I thought we'd talk about it. And it's really unfortunate that it's abbreviated AD. I'm sorry.
Yes.
I'm very sorry, Alan.
That's quite all right. I've covered this disease, and it's always a bit confusing.
First, herpes simplex viruses. Dickson, did you get it to work?
No.
Okay. Push the restart button or
The
whatever.
Wi-Fi, not the herpes.
Herpes simplex viruses types 1 and 2.
It's not going to do it.
These are rather large DNA viruses. They're enveloped, and they have a double-stranded DNA genome. How big is it offhand? Does anyone remember?
Ooh.
Wait a minute. I'll
Rich
go to
Con-
the internet and tell you.
Rich Condit, how big is it?
I'm going to guess 100 kilobases.
No, it's got to be bigger.
Probably too big. No?
Go to Viral Zone.
Yeah.
152 KB.
You got it there?
That was pretty good, Rich.
But anyway, most people get infected very early in life with herpes simplex type 1, which mostly causes the oral lesions. Typically, in your first few years of life, you get kissed by a parent, you get virus on your mucosal surface, or maybe a break in your skin, replicates. Eventually, you may or may not have symptoms. As a kid, you may get nothing, or you may have a sore. The virus.
I think most frequently you don't have symptoms.
Yeah, you don't initially. Virus then goes, if you have an infection in your oral area, goes into your trigeminal ganglia and becomes latent in the cell body, in the neuron, in the ganglia. And it becomes
Trigeminal ganglia, where's that?
Right at your jaw.
either side. There's one on each side. And it innervates your lips and your
One
nose.
of the 13
right?
cranial.
Yeah, it's just a little under your ear, right about where your jaw turns.
Ganglion is a bundle of nerves.
It is.
Right.
Yes.
Nerves that
And-
branch out from that and innervate your lips and your
There you go.
And we're just a bundle of nerves here.
Without them, you wouldn't have said that then, Rich.
And
or been able to.
so the virus goes from the mucosal surface there and becomes latent. And what latency means is that the viral genome is in the nucleus of the neuron as an episome, that is not integrated. And it's silent except for the production of one RNA called LAT, which stands for latency-associated transcript.
So it shuts everything down, but it leaves the lat on.
Right.
Very good.
Right.
Now, these are peripheral neurons,
Yes.
not central nervous system. Now, periodically, if you're under stress or if you go skiing and you get a lot of sunlight, these genomes reactivate and they begin to replicate and they make virus particles.
So stop right there just for a moment. Is
Yeah.
this then an antibody-controlled latency?
No, it's not.
So what's the stress doing?
Okay, we did a paper on that a while ago, and it's just fascinating. So the genome is chromatinized and silenced in the neuron. Methylated chromatin, it's not transcriptionally active. And when you're under stress, there's a neuronal kinase that's activated. It phosphorylates the chromatin. That partially de-represses the silencing, and then you get gene transcription. You guys remember that on an old TWiV?
Yeah,
I
this
think I
sounds
heard
familiar.
that.
It was very cool. What was it called? Anyway, it was a
What
good. It was a
are
good
you
episode.
transcribing then? Assume no herpes. What is the normal evolutionary
I don't know, but herpes will be
advantage of that?
I don't know. There must be a stress response. I don't know the advantage, but herpes
Stop
is
swearing or
transcribed.
something?
Herpes is transcribed. Make virus. It travels down from the trigeminal, and then you get a sore on your lip or your nose, wherever. If there is also HSV2, which is largely genital lesions, same idea. Okay. So, and this can happen over and over. You can get, some people shed without getting cold sores, but the virus sheds and you infect other people, and that's how it spreads to the new host.
Or shingles. Don't forget that.
Well, that's a different virus. It's a herpes virus. I'm sorry, Brianne, but it's a herpes. It's a zoster, varicella zoster.
But the same
idea.
Right?
Same idea. Yes.
Right.
Sorry.
Same process. So you had mentioned earlier that this happened in most people, and I just in terms of HSV1 infection. So I just looked at the CDC site, and they have some data as of February 2018, and it's 47.8% of people in the United States before the age of 49 have this virus.
That's a lot.
I thought it was more. I thought it was 85% seropositive.
I actually would have thought more as well.
I think that's low.
That's still, I think it's a lot.
So that's before the age of 45?
Yeah,
maybe before the age of 49.
Yeah, well, hang around for another 30 years.
Right.
So some people have a cold sore every 2 weeks and shed virus. Some people shed virus without a cold sore, but this is something that recurs throughout your life. Now, every now and then, in certain people with genetic lesions that have been studied, the virus goes the wrong way. It goes into your brain instead of to your lips, and you get herpes encephalitis.
That's a bad thing.
Which is a very serious disease. Yep. Well, any virus in your brain is not a good thing.
Yeah.
You're right.
I remember going to a herpes virus talk where they did basically what we used to do in the polio lab. They did an intracerebral inoculation to look at this process in the brain. Mm-hmm. And they had a major problem because all the titers they tried killed the mice immediately.
Mm-hmm.
And they had to go down to these, what we would consider ridiculously low titers of virus into the brain in order to get any kind of pathology they could even look at. Right. It's extremely deadly.
Yeah.
Herpes encephalitis is also a big problem in neonates. Children, in particular with HSV type 2, so children born of mothers who have active HSV type 2 infections are susceptible to encephalitis. It's treatable with antivirals, but it can be very, very nasty.
So this clinical trial is ongoing here at Columbia. You can go to clinicaltrials.gov. I'll put a link in the show notes. So, this is a government-funded clinical trial. And what they're doing here is the idea is that there's HSV in the brain, and this is connected to Alzheimer's, and we'll go into that in a moment. So they are taking people with Alzheimer's, and they're going to give them antivirals every day for 78 weeks, and they'll do a PET scan at the beginning and at the end and see if there's any, change in
78 weeks.
Yeah, it's a lot of antiviral.
Wow.
They're getting 2 to 4 grams a day.
Lordy.
Every day. Every day.
They're going to be virus free of everything. Well, of course, that doesn't work against all the viruses, but
so this when I first saw this, I thought, what is going on here? If you have herpes in your brain, you are in big trouble. So turns out there's a big literature. The first thing I did was I emailed David Knipe, who is a herpes virologist at Harvard Medical School. And I said, is there herpes virus in everyone's brain? And he said, yes, there are herpes simplex DNA sequences and transcripts in the central nervous system. It is unclear if it is replication competent or not. I would guess that abortive reactivation is causing inflammation and indirectly the neurodegeneration in Alzheimer's disease. I would say that at most herpes could contribute to AD. It's hard to say it's the main agent of AD. And he says there are antiviral trials for cognitive deficiencies, but the DNA synthesis inhibitors may not inhibit early enough to stop the inflammation. We would like to get something that blocks earlier. So the whole trial is predicated on the idea that there's infectious virus in the central nervous system and that the antiviral is going to hit it.
So could you repeat again for me, because I think I missed it, that every Alzheimer's disease person has herpes virus?
No, not every.
No, that's right. So what about the ones that don't?
So let's say at the onset, no one claims it's accounting for all Alzheimer's. It's just a fraction of the cases.
Right.
But we'll go over the data that implicates it, and you can see. Now, not too long ago, we talked about a study, TWiV 505, where they found HHV6 and 7 transcripts in Alzheimer's brains. No virus, just transcripts. The preponderant viruses were not herpes simplex, but 2 different herpes viruses. And so maybe if a virus gets in the brain, it could cause damage and trigger AD too. And go ahead.
What evidence do we have that valacyclovir crosses the blood-brain barrier?
It is used to treat encephalitis.
Oh, it is. Okay, so
yes.
it does.
So it does get in there, yeah.
Okay.
Now, this has been in the news recently. You can Google herpes and Alzheimer's, and you'll get all new news. I saw a BBC headline the other day, and I mentioned this to Brianne a couple of weeks ago. New evidence implicates herpes simplex and Alzheimer's, so I was wondering what that's all about. So I decided to do this episode. We should do this episode and talk about it. And a review article has come out in, Frontiers in Aging Neuroscience. That's a journal.
Which is a funny title for a journal.
And the title of the article
They publish old papers or what?
Right.
Corroboration of a major role for herpes simplex virus type 1 in Alzheimer's disease. Okay. And so that has spurred the news reports. And the summary of this is that, and in fact, this, you can find quite a few papers on this association of herpes and Alzheimer's. It doesn't mean that they're all right, okay, just because there are a lot of papers. But a lot of people say, there are over 150 papers on this topic. Yeah, but if there are 150 bad papers, that doesn't help you. But
this is true.
Ruth Itzhaki is at the University of Oxford. She has done a lot of the work in this field. She wrote this review article, and she says, our theory is that in people with a specific isoform of apolipoprotein E called epsilon 4, and in these people, you have herpes simplex reactivation more frequently. It's more harmful in the brain, and you get damage that culminates in the development of Alzheimer's. Okay, so you look way back in the 80s, people started looking for herpes viruses in the brain by PCR. You take brain tissue of controls and Alzheimer's patients.
Why is that the wrong thing to do?
What's that? PCR?
PCR. Exactly right.
Well, PCR is picking up DNA.
There's no plaque assays.
Yeah, you're right. There's no plaque assays. So you can find a couple of studies where they have looked in Alzheimer's patients and normal controls. There's one from 91. They found the viral TK DNA by PCR in all brains of 8 Alzheimer's patients and 6 normal controls. Okay. And then there was another paper where they looked in normal individuals, i.e. non-Alzheimer's, and they found herpes simplex in 28% of their normal tissues. And they also found other herpes viruses as well. But again, these are pieces of the genome. It's not infectious DNA. Exactly. And then there have been some other studies. There have been epidemiological studies that make a connection between being herpes simplex seropositive and developing Alzheimer's, but those are associations, of course. They don't prove causation. They have, as I mentioned, this apolipoprotein E epsilon 4 seems to confer a high risk of Alzheimer's disease. It also is a high risk factor for cold sores, implicating both herpes and that as well. They can make, as we'll see in a paper we're going to talk about later, when you infect cells with herpes simplex 1, you induce amyloid-beta accumulation. And amyloid-beta, of course, is what accumulates in the brains of AD patients and causes plaques, and it's thought to be one of the contributing factors in addition to other things.
Right.
Well,
so there's this debate ongoing, and I'm not sure exactly where it stands at the moment, but a lot of discussion in the Alzheimer's disease field surrounds what is causative and what is an effect.
Right. Right.
So there's been a lot of study and question about amyloid-beta and these amyloid plaques that form, not to be confused with viral plaques, although I guess, So yeah, and or in fact, you in definitively diagnosing Alzheimer's disease, you post-mortem, you look at the brain and you assay for these plaques. So I guess they do plaque assays too.
These are big, ugly tangles, of aggregated protein.
Big piles of protein. in some respects, people have hypothesized that this is in some ways parallel to prion diseases. Because it seems that when you get these plaques forming, they then crystallize and nucleate more plaque formation. So a lot of discussion has been around, well, okay, that's what's causing the neurological damage, but there's also the possibility that the neurological damage is happening and then the plaques are growing as a result of that. And this idea that amyloid beta is possibly an antimicrobial response of the cell would maybe feed into that.
That's right. That's right.
There's another protein involved called tau protein that some people think is more likely to be, and we're going to talk, I think, a little bit about hyperphosphorylated tau, which is also found in these brains. And some people have jokingly suggested that you could divide Alzheimer's researchers into the people who believe in amyloid beta protein or beta amyloid protein, and those who believe in tau. So you could call them the Baptists and the Taoists.
Right.
I did not come up with that.
Remember, science is not a belief system.
That's right. But there are different schools of thought about exactly what the roles of these proteins are.
Go ahead, Brianne.
I think that one other thing that is important to realize, I have a colleague here who works on Alzheimer's, and so I've learned a fair amount of Alzheimer's biology from him. And one thing that I think is also really important to realize is there actually seem to be slightly different subtypes of Alzheimer's in terms of things like early onset or late onset. And so one reason why some of this might not be able to explain all types of Alzheimer's, one reason for some of this variation might be that there are more subtypes of Alzheimer's than we might realize.
Right.
And one of the aspects of this amyloid beta is that there's a precursor that is cleaved by, beta secretase to form the peptides, which then form the plaques, which deposit to form the plaques. And in fact,
there is a human genetic model, people with Down syndrome all develop Alzheimer's disease.
And they have increased
all of them?
Yes. This is a standard progression in Down syndrome. As those people age, they get beta amyloid deposition and development. I think it's 100%. It's
and what's just they're trisomy 21.
Trisomy 21.
So is there any gene involved that's amplified there?
I haven't really delved into that literature. That's we've just about reached the extent of my knowledge on it. It's just interesting. Yeah. There's this this population that genetically they're obviously very predisposed to this, and we don't know why.
If they're convinced that this beta amyloid protein is the cause of
they're not. See, they're not.
No, but there's a way to show that, though, isn't there? Why don't they use interferon RNAs and try to block the synthesis?
That gets we're in humans here. Now there are mouse models. We'll get to that.
Okay.
But,
all right.
So the in herpes infected cell cultures, you get an increase in amyloid beta. You get activation of the protease that cleaves it. I mean, these are all interesting and could in fact be significant, but we don't know. In infected cells, you get hyperphosphorylated tau, which leads to its aggregation, I think. Right, Alan?
I think that's right, yeah.
I think so, too.
Another interesting finding in amyloid plaques in people, in the plaque you find herpes simplex DNA right in the plaque. Of course, it's not the whole virus or it's not infectious, but it's DNA. In cell cultures where you get increase in amyloid beta and phosphorylated tau caused by herpes simplex virus, of course, the antiviral will inhibit that. That's not surprising. What else? Inhibitors of amyloid beta and phosphorylated tau will reduce those levels in viral infected cell cultures. So there are all these links, and the final one, of course, is that, and we'll get to a paper that talks about that, as Alan mentioned, and we've mentioned before. There's growing evidence that amyloid beta deposition is actually an innate immune response to microbial infection of various sorts, to kind of encapsulate the bacteria, even fungi, viruses, and prevent them from infecting cells. So we have a paper that we actually mentioned when we talked about the HHV6 paper a while ago that looks at that in cells and in mice.
I think they were published back to back.
Yeah, they were. You mentioned that, and so I thought we could go over that in a little detail. Now, what has really triggered this reemergence of interest in this is that a couple of papers were just published from Taiwan in the past year. Now, Taiwan's interesting because 99.9% of the population is enrolled in a national health insurance research database. And you can, of course, you get the permission. You can mine this for information. And so what they did is ask, can we find people who have been treated with anti-herpes simplex antivirals, and let's look at the incidence of Alzheimer's in them. So they did that, and the one paper which I think is the most striking, it's anti-herpetic medications and reduced risk of dementia in patients with herpes simplex virus infections, a nationwide population-based cohort study. So remember, this is just looking at data. There's no intervention at all. And it is using senile dementia, of which Alzheimer's is a major cause. So they had 8,300 subjects, 50 years of age or older in 2000, diagnosed with herpes simplex 1 or 2.
And also it says newly diagnosed. So does that mean that they became infected recently?
Yeah, I suppose.
Was what I wondered.
I don't know.
I think it's highly unlikely that they became infected recently. At least with all of them. Since this is such an incredibly common thing and people don't go to the doctor for a cold sore normally, I'm going to presume that these are people who had some reason why it was particularly concerning to them. Either it was reactivating very frequently, or there was some issue that caused them to get treated for this, because most people don't bother to treat it. Right.
Yeah. I think
so.
You're absolutely right about that.
Out of the population of Thailand, essentially everybody in Thailand, and this is not a small country, population-wise, or Taiwan, sorry, not a small country population-wise.
No.
They had 8,300 people who had been diagnosed and underwent treatment, and then they had their controls were 33,400 people who didn't.
So in this study, the 25,000 controls, no HSV, and they followed them over 10 years, which they can do in the health record, looking for dementia. Risk of developing dementia in the herpes group was 2.56-fold greater than the control group who didn't have herpes infection. Okay, fine. And then a group of the herpes patients, subgroup 7,200, were treated with antivirals of all sorts, different antivirals. Cyclovir, valacyclovir, etc.
Right. So, sorry, I think I misstated. The 8,300 was the people diagnosed, and then the subset of those 7,000 something. Right.
And they had a 10-fold decrease in the incidence of senile dementia compared to the untreated people.
That's a big difference.
Yes.
You have to sit closer to the mic, please.
That's a big difference.
Yeah, it is a big difference. So it's clear that there's some association between the drugs and the dementia, yes. So here are the numbers. In the subgroup with the antivirals, 419 or 5.8% developed dementia over the next 10 years. In the subgroup without the antivirals, 325, which was 28.3% developed dementia. So the authors say the risk of senile dementia is much greater in those who are infected with herpes, and antivirals cause a dramatic decrease. But they also say, we don't have any serology,
right? So
we don't know who's actually infected. And I would say that you really would need to know if the antivirals decreased the presence of virus or not, right? Because as Alan made a note, an antiviral is a drug. It could do other things. And of course, there could be other confounding factors, right, in this study. Besides taking antivirals, maybe there was something else in common to them that you didn't tease out in these health records that is making a difference. So I think it's an association. It's intriguing, but I'm not sure it proves anything. Okay, now at this point, let's read an email that Islam said.
I think it does prove that Taiwan is way ahead of a lot of other countries, including us, on
yes,
putting these databases together and maintaining that healthcare system.
Yeah, that's true.
Because I mean, what an awesome resource!
That's great.
Totally.
It sounds like Iceland.
I don't know if you remember, we did a paper on TWiV a while ago on the new varicella zoster vaccine, and they compared it to, oh, no, it wasn't the new one. It's on the efficacy of the old one. And they mined Kaiser Permanente health records.
Right.
Hundreds of thousands of health records where they could say, look, the thing wears off. So, yes, the digitized health records are very useful.
Most of them are in the hands of the insurance companies, however.
Yes, but shout out to the insurance companies, though. They have actually been pretty good about working with researchers to a lot of it is that they have to obviously protect the privacy of those data, but there are now protocols in place that allow a lot of research to get done on that. It's not quite as convenient as a single national health insurance database that Taiwan has. But yes, if you work with Kaiser Permanente or a couple of the other big insurers or the VA, you can get these kinds of data sets.
So how secure is the Taiwan data set?
I don't know.
How what?
Secure. I mean, do people in Taiwan know each other's health
No,
records?
I'm sure you can't.
Well, in that case, they must have a way of protecting the individual. So why don't we do that?
Well, no, we do. So
no. Why don't all the insurance companies do that and make a big giant database, government sponsored,
okay, so there is,
that everyone can access?
That would make too much sense.
No. Hold on. This kind of exists. Not every insurance company is on it, but there is a consortium. I forget the name of it, but it's set up in a way that the insurance companies have their databases, and those are protected from access, and they've got all the firewalls and everything. What researchers can do is they put together their queries for the databases and submit their query, and then the database, the insurance company will return the results of that, like this many patients match this criterion, such and such, without revealing any more information than the query requires. Mm-hmm. So all the information stays with the insurance company, and only the answer to the query comes back. And the query can't be, does Joe Smith have cancer? The query has to be formulated in statistical terms. And so there are protocols in place that can actually be used to allow this type of research without breaching anyone's medical records.
Hey, Rich, can you read Islam's letter, please?
Sure. Dear Vincent, near the end of TWiV 517, there was a mention of HSV-1 and Alzheimer's, AD. You expressed a disagreement with a news headline referring to a mounting evidence for an HSV-1 involvement in AD. As you know, I work on antivirals, and the recent 2 papers published in Neuron last July, discussed on TWiV 505, sparked several discussions amongst my colleagues about the possible role of herpes virus infection in AD and whether antiherpetics could fit into the picture. Since then, I have been diving into the literature to try and understand what this is all about. I was stunned by the body of data supporting an infectious cause for AD. I think you might be already familiar with this hypothesis, but for the uninitiated TWiV listeners, please allow me to give some background. For quite some time, amyloid-beta has been considered as a functionless catabolic byproduct. However, numerous studies suggested that it is an innate antimicrobial peptide which exerts its activity via aggregation into plaques, a characteristic feature of AD. This is commonly referred to in the literature as the bioflocculant hypothesis, where amyloid-beta oligomers agglutinate pathogenic agents in the brain, and by so doing, they would limit their spread and prepare them for phagocytosis. Indeed, synthetic amyloid-beta exhibited potent in vitro antimicrobial activity against some viruses such as HSV-1 and influenza, but not adenovirus, which led some authors to conclude that amyloid-beta interferes with fusion with the target cell membrane. Actually, timeout. Given the snippet we're going to come up on, it makes sense that you would get interference with HSV-1 and influenza and not adenovirus, because the amyloid sticks to carbohydrate. Okay. And so envelope viruses like HSV1 and influenza that have glycoproteins might bind amyloid-beta, and adenovirus, which wouldn't.
Yep.
Furthermore, in vivo data by Kumar et al. Suggested that amyloid-beta protects against fungal and bacterial infections in mouse, nematode, and cell culture models of AD. Also, clinical trials of anti-amyloid-beta therapies in humans frequently reported adverse effects, suggesting a dual protective damaging role for amyloid-beta. With regard to HSV1, I'm actually inclined to agree with the news headline that you didn't like. After consulting with the body of literature that I came across, I think there's considerable evidence supporting HSV1's involvement in the pathogenesis of AD. I'm attaching a few papers and reviews for your reference, but will summarize some of the findings that caught my attention. I would particularly invite you to check out papers by Wozniak and Isaki. In AD brains, HSV1 DNA co-localized and was found to be associated with amyloid plaques by in situ PCR. I know that you're going to say that this is a latent latent DNA, not virus. Surely enough, anti-HSV1 IgM antibodies, indicative of primary or reactivated infection, in CSF significantly increased the risk of developing AD, suggesting that reactivated virus played a significant role. Several meta-analyses concluded that HSV1 and other infectious agents are associated with increased AD risk. Antiviral treatments greatly reduced amyloid plaque accumulation in HSV1 infected cells. In HSV1 induced encephalitis, HSE, the main regions affected, the frontal and temporal cortices, are those showing the main pathological changes in AD. It seems to me that repeated reactivation of HSV1 in elderly brains, probably as a result of immunosuppression, causes gradual cumulative damage and eventually contributes to the development of AD via the formation of amyloid plaques. I am actually more inclined to believe that HSV1 is not the sole agent, and other neurotropic pathogens could be involved as well. You've promised to dedicate a TWiV episode to this intriguing topic, and I look forward to a hot discussion among the TWiV teams. Mm-hmm. Thanks very much for reading my long email. Best regards, Islam. So here you are, Islam. We're right in the middle of it.
So,
yes, we are.
So the presence of antibodies does not indicate virus infection. Let me remind you that we have several inactivated virus vaccines, virus-like particle vaccines, that give rise to antibodies. So viral proteins could just as well give rise to these CSF antibodies. So I looked up the paper. There's a single paper, the title is Productive Herpes Simplex Virus in Brain of Elderly Normal Subjects and Alzheimer's Disease Patients. And they looked for antibodies in the CSF. So I think the title is not right, Productive Herpes Simplex Virus Infection, because, as I said, proteins could give rise to the same antibodies. They use an ELISA to look for IgG. They find, first of all, the titers seem to be low. They find HSV1 IgG in 14 out of 27 Alzheimer's patients and 9 out of 13 age-matched normals. And they claim that this shows that these people have been infected, but I would say it's kind of a limited study. I haven't seen it done elsewhere, and it doesn't mean that there's infectious virus there. And it's IgG. I think it's not IgM, but that's okay.
Also, while we're on the subject of the immune system, there's another thread in Alzheimer's to tie in here, and Brianne has probably heard a lot more about this than I have, but there's some discussion, or there has been some discussion, about whether Alzheimer's disease could be considered type 3 diabetes.
That is actually exactly what my colleague works on.
Okay.
So, yes, I've heard quite a bit about this.
All right. Yeah. So, and a lot of that ties into how the immunology of Alzheimer's disease is strikingly parallel to the immunology of diabetes. And it's possible that what we're seeing in both cases is sort of misaligned inflammatory response that's either driven by diet in the case of type 2 or driven by genetics in the case of type 1 or driven by question mark in the case of Alzheimer's disease that leads to these pathological changes, and you see insulin changes and such. And I'll let Brianne take it from there.
So yes. So there is actually both kind of the biological evidence that you mentioned and also a fair amount of epidemiological evidence showing relationships between diabetes and Alzheimer's disease patients, such that some people have this idea that Alzheimer's is a type 3 diabetes, possibly including things like changes in responsiveness to sugars or to insulin in brain cells and things of that nature. And that also might be tied in with changes in signaling in neurons that are involved in oxidative stress and oxidative damage. And whenever I see a lot of these data, I think to myself, okay, so there has been epidemiological data and biological data showing relationships with Alzheimer's and inflammation, between diabetes and inflammation, between oxidative stress and inflammation. And so I wonder whether all of these are tied to responses to inflammation, whether it's people having some sort of genetic predisposition or something like the viral infection leading to additional inflammation. And I really like where Islam said that he's inclined to believe that other pathogens could be involved. And originally you also mentioned with David Knipe's comments, some ideas about inflammation potentially being there, that he thinks that the abortive reactivation was causing inflammation and then indirectly the neurodegeneration. And from what I've seen of all of these theories, my thought is it sounds as though there's some trigger leading to inflammation, and the inflammation is the unifying factor.
And I just want to point out, you can't tell which way the train went by looking at the tracks. So it's possible that we have this backwards.
I love these rural virology statements.
that in fact, it's even possible you've got an inflammatory process going on, and as a result of that, HSV is partially reactivated, or some genes are transcribed, or something causes its DNA to get someplace like the brain where it wouldn't normally be. And HSV didn't start the inflammation, but it's there because of the inflammation. And what we're really be getting at is the inflammation, in which case, again, it's not necessarily HSV causing Alzheimer's disease. It could be it's there because all this other stuff's going on.
Exactly.
So how would the amyloid-beta fit into all of this?
One might imagine that if amyloid-beta is an antimicrobial peptide, that changes in inflammatory responses would change the production of that antimicrobial peptide.
Okay.
And it may be that if you've got, I'm just, shooting off the cuff here, but I could imagine something like amyloid beta is supposed to be expressed and tangle up virus, and if there's not virus for it to tangle up, it tangles up itself. And maybe you've turned on this response inappropriately or excessively, and somehow that's causing the amyloid to accumulate.
So Brianne, when you say inflammation, what's in your head?
No pun intended.
It's a tricky question for me to answer. How much time have we got here? But really, I'm thinking about sort of innate immune cytokines and other types of sort of the early inflammatory response. So I'm thinking maybe about cytokine production, but I probably use that word a little more carefully when I'm thinking about my research and the cytokines I measure, as opposed to when I'm talking about this, where there's a lot of other types of data that are across the immune system that have been seen.
Okay.
So my main problem with this is the idea that there's infectious virus in the brain. I like David Knipe's idea that these are abortive reactivations, because I just do not understand how you could have multiple, let's say the virus is latent in your brain ganglia. You could have multiple reactivations in the production of infectious virus and you're not getting encephalitis. I just don't buy that.
Yeah, so this to me is really an important point, and I think the abortive reactivation is a good way to think about it. This latency thing, in my mind, is not black and white. It's not all on, all off. Okay. And, my sense is that, maybe I'll get email about this, but my sense is that in the latently infected cell or during latency, the virus may be sort of, trying itself out all the time, kind of peeking its head out the door, a little bit of reactivation. Okay. Maybe enough even to replicate a little bit of DNA and maybe make some, gene products from the whole virus, maybe even make a little bit of virus, but because of the immune response or something like that, it gets shut down. Okay. Enough. So it's just flying under the radar. Occasionally you get a reactivation that's full blown and you're making virus, and then maybe you could get encephalitis or a cold sore or whatever. But I think that you could have a lot of low level activity that would escape our detection via antibodies and DNA replication, et cetera, but nevertheless might be enough to trigger the sorts of pathologic responses we're talking about, or to be sensitive to antivirals.
Well, that's the thing that I'm wrestling with. So if it's abortive, then an antiviral's not going to have any effect. Antivirals are for the acute phase. You get a cold sore, you take valacyclovir, the cold sore goes away, you've cleared the virus. If you've got these latent infections in the brain, I don't know how the antiviral is going to work unless, as you say, Rich, you get these mini reactivations, a little bit of virus, the antiviral knocks it down.
Yeah.
And otherwise, it comes up and it's cleared, it comes up and it's cleared, and eventually the accumulated damage causes this plaque formation. But that is just contrary to what I have been thinking for years, that you have a little herpes in the brain, it is a disaster. So now we're saying a little is okay. So that's why I have trouble with this. I don't know how to interpret the Taiwan data. They don't make a lot of sense to me. And the clinical trial they're doing now, they're going to treat people for 2 years. What if you stop? The latent DNA is still going to be there because these antivirals do not take away the latently infected cells. Are they going to get Alzheimer's back again? Or do you have to take antivirals for the rest of your life? So that's why I have trouble with this whole idea. I can't explain the Taiwanese results, but I don't see that an antiviral that blocks DNA replication is really going to have an effect on this.
So from an epidemiological standpoint, forget virology or microbiology in general, just look around the world and ask the question, if you had to put a red dot for every 1 in 10,000 cases of Alzheimer's disease, which country would have the most red dots?
The one with the oldest population?
You think?
I would assume so.
Well, that's an assumption. That's an interesting assumption, but I bet you that study's been done already.
I'm Googling.
This is just
Alzheimer's is most common in Western Europe.
Western Europe.
And North America is close behind. It's least prevalent in sub-Saharan Africa. Probably because people die.
Expectancy.
No, but if you go to Cuba, they have the longest female average lifespan of any place in the world, 83 or 84 years old, something like this. I wonder if they have any Alzheimer's.
Probably.
Yeah, it's some, but I wonder what the rate is. And there are some places in Russia where people live a long time, too. That, yogurt, all that stuff.
Right.
Really, I mean, there are some countries like Bhutan, if you live past the age of 5, you're probably going to live to the age of 90. So I wonder about these isolated populations where you have, the infectious disease rates are different there than they are anywhere else. Mm-hmm. You could say, well, in that case, this is excluding this because this doesn't exist here, that sort of thing.
So I think that it really makes a lot of sense that you would get partial reactivation, viral proteins. That's why you get antibodies in the CSF. And then you have an innate response, which includes the production of beta amyloid and the deposition. And there we don't have experiment. People look at infectious virus and they say it induces amyloid beta. What about viral proteins? Is that enough to do it? I think those would be experiments worth looking at. Because you might consider making a new antiviral that acts earlier in the infectious cycle, right?
Or acts against proteins.
Mm-hmm.
This is
so a candidate would be its viral protein VP16, which is needed for the very first transcription.
RNA would do
you could make you could make an inhibitor of that.
Yeah.
Well, you'd have to get it in the CNS, I guess. But so this current clinical trial, I would be surprised if it shows an effect of any kind on these individuals, because it just makes no sense to me in terms of infectious virus. I can't explain the Taiwan results, but I just don't think this one's going to work.
Wouldn't we expect that something that would be very helpful would be a vaccine?
Oh, yeah, for sure. Because,
prevented the latent virus in the first place.
Yeah, because, I mean, herpes is a good candidate for a vaccine because it causes all sorts of problems. And, cold sores in some people are really an issue.
Absolutely.
Every 2 weeks.
Yeah.
It's very bad.
You're right.
So I think a vaccine would be great. And then, then you do population studies. Does that decrease at least some of the Alzheimer's incidence, right? I just don't think giving 2 years of 4 grams a day of antivirals is a particularly good idea. What you think?
So I had a different take on this. I agree with you on the virology that we really don't know what's going on mechanistically here in Alzheimer's disease or the herpes aspect of it. We don't know what's cause, what's effect, whether the virus is a spurious finding or involved in some way or involved in what way. And I don't buy the idea that the antivirals are going to, that the Taiwan study is explained by the antivirals stopping the virus, and that's what stopped the dementia from developing. Because these are replication inhibitors, as you said. That's not, and we know the virus is not up to that stage because otherwise these people would just be dead.
Exactly.
So what I do see, though, from a medicine and science policy standpoint is you have all these papers in literature, all of these suggestive things that suggest roles for HSV in Alzheimer's disease. And a lot of it really points to, this is something we need to look at. Then you have the study from Taiwan that came out in April. That's a vast population study that shows what I would consider pretty impressive results, right? I mean,
for sure.
Tenfold reduction in senile dementia for people who were on antivirals. That's the kind of finding that a lot of doctors who treat, gerontologists, people who are treating the elderly, are going to look at and say, I've got this guy, he's got mild cognitive impairment, he's probably going to progress to Alzheimer's, or he is progressing to Alzheimer's. I'd really like to be able to do something for him. I see this paper where it's suggestive that antivirals might be useful. I'm going to go ahead and write a script off-label for valacyclovir.
Sure.
That's going to happen.
Yeah.
So
I agree.
I'm actually in favor of someone doing a proper study like this one, like this phase 2 study at Columbia, and asking the question straight out in a proper placebo-controlled fashion. Does this work?
I agree. You could not, based on the Taiwan study, you have to do this clinical trial. I agree with that. Right. I'm just going to be surprised if it has any effect.
I will also be surprised, but this is a disease for which there's currently nothing.
Nothing. I totally agree. People are dying to have.
And if this works, and these drugs are, I think, usually fairly well tolerated. I mean, these are high doses and you do worry about resistance arising. But if this gets some kind of a benefit, then we can start teasing apart how the heck it works, because it's probably not, working on the basis that people hypothesized to get us at this point.
Right.
But that could be very promising.
I think Islam, if you want to go after this, go after a VP16 inhibitor.
Yes.
You can make a very nice assay for VP16 activation of viral promoters, and you can screen compounds for inhibitors. And I think that's the way to go, because it just doesn't make sense to go for a later inhibitor.
Unless you want to take on the big challenge and develop a vaccine.
And then I would hook up with David Knipe, who knows an awful lot about this and would be a good colleague, and he's right there in Massachusetts.
There you go.
August 2022. That's when this is going to be done.
Yep.
Interesting.
So that'll be TWiV.
Actually, it's only another 150 episodes, right?
Yeah.
Listen,
We'll cover it.
we'll be there.
Perfect.
All right, let's briefly talk about this paper. It's published in Neuron. It's a report. So, Alzheimer's disease-associated beta amyloid is rapidly seeded by herpes viridae to protect against brain infection.
Now, a report in Neuron should really be called a signal.
It should be. Yeah, you're right. This comes from Massachusetts General Hospital and Harvard Medical School. And Kumar is one of the authors who was mentioned by Islam. And the first author is Eimer, and the last author is Moir, M-O-I-R, sorry. And there is an author name here that is someone I never met, but whose name I have seen for 40 years, and who I think has the coolest name, Xandra O. Brakefield. Isn't that a great name? Xandrao, Brake with a Breake. She is at Mass General, and she's been in this field for years, and I always thought the name was cool. So here they're looking at amyloid beta and its effect on herpes simplex virus, both in cells and in mice.
Testing the notion that it has antimicrobial effects.
That's right. And they have they have this mouse model where they're transgenic for
human amyloid beta?
Human amyloid beta.
With a mutation, or is it just wild type?
No, they got mutants. As a matter of fact, they got several different mutant alleles. They got 5 different pre-amyloid proteins, I think, that come from different mutant backgrounds. So these are prone to aggregate. Right. And they've also put in a gene for presenilin, okay, which is it's responsible for the cleavage, or it's a subunit of the enzyme that does the cleavage to get you from the pre-amyloid protein to the amyloid protein. Okay. So these mice are really set up for disaster.
So my understanding is as they age, they develop beta amyloid deposits, right?
That's right.
So like over 4 months, but the young mice, 5 to 6 weeks, they're okay. And they make this human amyloid beta, and they infect them intracerebrally with herpes simplex type 1. And they look for survival. The ones that are transgenic have increased survival compared to wild type mice by virtue of having amyloid beta in the brain. Increased survival, different weight loss compared to litternates. So they say, look, this is protective in this particular model. I would agree that the results look like it's protective, right?
Sure.
Mm-hmm. Then they go to cell culture. Can amyloid beta protect against infection with herpes in cell? And the answer is yes. If you have amyloid beta produced, the viral production is attenuated. But it's got to be in the cell. It doesn't work if you pre-incubate the cell with the amyloid beta. It's got to be in the cell itself. Okay, so how is this working? They look at binding of amyloid beta to virus particles using a ELISA assay, basically. They put the virus on the plastic and they say, can amyloid beta bind? And the answer is yes, it binds. And it binds the virus, it binds simplex, it binds HHV6A and 6B. And they do the controls with scrambled peptides and immunodepleted culture. It looks like it's binding. And it looks like Rich said earlier, it's binding to carbohydrates because the binding is inhibited by a yeast polysaccharide mannan that will bind to the carbohydrate. And they further look at the carbohydrates that are involved there. So the amyloid beta is binding the herpes virus glycoproteins, specifically the carbohydrate moieties. And that's probably how it's inhibiting, although they don't show that. I could think of a number of experiments to do that, right? Mm-hmm. But they don't do that exactly. As we mentioned, these amyloid beta make fibrils. And they ask, is this part of the way that it's blocking virus infection? So they look at the virus with the amyloid-beta peptide under the electron microscope, transmission electron micrographs, and they see fibrillar structures attached to the viral membranes. And these happen within 15 minutes. So you get this network of fibers, and they can show that it's the amyloid protein. They do gold-labeled antibodies, and they can see these binding to it. And in cell cultures, they can see the deposition of these fibers in after infection.
And one of the most interesting things about that to me is that it's almost as if the infection actually it induces the deposition of the fibers.
That's right.
And controls that are uninfected at this particular period of time in the culture, you don't see the fibrils, but in the infected ones you do.
Yeah. And since the glycoprotein can bind these amyloid beta proteins, I think it would be interesting to know if that alone, just the glycoproteins in the absence of virus, would that be enough to bind and cause fiber structures to form?
Right, because maybe it's just that that carbohydrate is sort of nucleating the structure formation.
Right.
You might not need the whole particle, right? Which I would feel better about, knowing that not everyone has infectious herpes viruses in their brains, like you, Dickson. You feel okay?
What did you say?
You can't feel okay?
Why would you ask? Can't you see that I'm
I wish
asleep?
there were ways.
How dare you wake me up?
I wish there were ways to look in people's brains non-invasively for viruses, infectious viruses. There is no way.
Yeah.
There's no way to do it. Someday someone will figure it out, a smart young person. And now,
did we say that in these cases where we're seeing in culture or mouse brains the appearance of these fibrils, that they co-localize with, what are they looking at? Herpes virus glycoprotein.
Yes.
So they're in the same place.
They're in the same place. Mm-hmm. Yeah, and they also
consistent with the aggregation of the virus particle.
Yeah, they also look in mice. They infect mice, and they ask, does infection seed the deposition? And normal mice don't have these deposits, but you see them within 48 hours after infection. Mm-hmm. And they do immunofluorescence. And as Rich said, even in mice, they co-localize the virus with the amyloid beta. So, and they say these deposits resemble the classical AMP, antimicrobial peptide mechanism of viral entrapment by fibrils. So as we mentioned, these are probably an antiviral mechanism. Although, Brianne, isn't it weird that it would be so active in the brain? Most of them are attenuated, right?
It is, yeah. The brain, we always talk about as the immune privileged site, where so many responses are attenuated. So it is interesting to see such an active response in the brain.
Yeah, because I always say, there are lots of barriers to getting into the brain. Mm-hmm. But some viruses can get in, polio, measles, West Nile, and when they do, it's a mess, right?
Yeah.
And
for the most part,
because interferon responses are attenuated. So, but there are exceptions, yeah. And I just remember mumps, in half of kids who get mumps, it turns out that those kids also have virus in their brain.
And chicken pox.
But they're fine, but they're okay. Maybe they're not okay. I don't know.
And there's the whole story with JC virus.
Yes.
Which, was in the brains of people. We didn't really know about it until other patients were treated with T-cell trafficking inhibitor, so T cells couldn't go to their brains anymore to prevent multiple sclerosis. And suddenly this virus started causing all sorts of problems that we never even knew was really in the brain beforehand.
Yep.
Right.
So that's basically this paper showing that herpes causes that it binds to amyloid beta. You get the deposition of fibrils in cells and in mice. And I think it goes beyond herpes. Other viruses could do this as well.
Other viruses, other microorganisms, fungi, bacteria.
Yeah. Exactly.
And also, when we talk about the brain being immunologically privileged, that's not immunologically inactive. There's a whole parallel set of things that goes on there with the oligodendrocytes, the microglia, and there's an immune response there. It's just different from what you see in the body.
Right. In fact, they use similar signaling molecules for the immune system. Yes. So that's like direct connection.
Yeah, that's next week's lectures in my immunology class.
I'm amazed that someone hasn't done, some really good studies on biofeedback to see whether or not you can think yourself into an immune state. Because you've got the power for it. You've connected to all the immune system by nerves. What the hell is a nerve doing in a lymph node?
Isn't that amazing?
What is it doing there?
It's controlling it.
Of course it is. Now how?
There are studies that, correlate mental state with immune status.
Understood.
Mm-hmm.
Understood.
And there are lots of stories about neurotransmitters affecting immune cells.
Totally.
Right.
We had a seminar here a while ago, guys, showing how brain activities can control lymph node activities. Oh, amazing. I wish I remembered his name or her name. I can't remember. That we should do something like that. All right, let's do one round of email. First one is from Matilda, who writes, Dear TWiVomers, weather is humid as usual, 82F, 28C in New Orleans. Hey, that's where you just were.
It's true. It's my hometown. It's my hometown, boy.
And apparently Rich was there. Rich Condit asked me this weekend if I had written to TWiV, and I embarrassingly had to say not yet. Decided to change that as I'd like to share TWiV's impact on my life. 2 summers ago, I was in the midst of choosing where I wanted to pursue grad school. As I was home in Sweden and helping to mow the large lawn, I used that time to listen to TWiV and TWiM. I can associate Zika epidemic updates, Hep C, and polyoma episodes with certain acres of grass that I mowed. It was at that point I decided I wanted to pursue research of viral host relationships for my PhD. Nice. Fast forward to now, I now study determinants of congenital cytomegalovirus transmission in a rhesus macaque model at Tulane National Primate Research Center in Covington, Louisiana. I recommended Rich Condit for Tulane's annual biomedical sciences retreat, so hence I got the honor to meet one of the TWiVos in person.
Nice.
He held an amazing keynote talk on his experience as a scientist, a constant fan of nature, and values which aided his career. Many people told me right away that he did a great job and was perfect speaker for the occasion. Thanks again, Rich. I hope the World War II Museum was good.
It was awesome.
Now that I'll soon be done with my classes, my 1-hour commute from New Orleans to the Primate Center will be filled with TWiV episodes instead of lectures. I can't overstate how much I am looking forward to that. P.S. My mentor is an immunologist, and I am trying to get her into listening to Immune. Attached is a proof of Rich's useful absentee from the last recording. She sends a picture of Rich giving his talk, which is entitled Accidental Virologist. And Matilda is a PhD student in her second year at Tulane.
Awesome. Right. I'm glad we helped you to get to the monkey house, Matilda.
Rich, you have a shout out.
I just want to shout out to not only to Matilda, but Joe Hoffman, who is a graduate student who was my sort of the point man in hosting me, and Zilkia Lozano, who was a staff member who did a lot of work in support of this thing. And I just had a great time. I think the Tulane Biomedical Sciences program is a really great program, and it was a really nice retreat, and I had a wonderful time. So thanks to all of them, and hello.
Now we know why he wasn't here on TWiV.
Yeah, we do.
Okay. Alan, can you take the next one?
Sure. Jake writes, hello. I'd love to say I'm a longtime fan, but I actually just started listening last week due to a suggestion by my microbiology lecturer. I really enjoyed last week's episode, and you're all great personalities. That's a nice way of putting it. Thank you. The one thing I wanted to say is please look into the glyphosate paper more. I've read it myself, and it's rather questionable. A few of the papers it cites itself are pretty questionable, too, and already debunked, so to say. I'm no Monsanto fan, but this paper is so fatally, fundamentally flawed, even in its foundations. And he links to a YouTube summary of it. Says, I'll be listening next week. Thanks, Jake, who's a second-year biomedical sciences student at Northumbria University in the UK. And you have a link to TWiM?
Yeah, we talked about the paper on TWiM. Michael Schmidt wanted to talk about it.
Okay.
It has to do with the microbiome of bees. So they, first of all, the lab, Nancy Moran, she's in Austin, Rich.
Okay.
Did you know that? She's at the university.
Nope.
So she's a well-known endosymbioticist. Is that the right word?
Okay.
And she's really, really good. And what they do in this paper is they feed bees glyphosate, and then they look at their microbiome, and they see it's altered. And they don't they feed bees that are in hives, so they're flying all over the place and doing their normal stuff, and they don't even get most of them back because they fly off and they can't or whatever. And so the microbiome is altered, although we don't know what that's going to do. But they do one experiment where they challenge them with a bacterial pathogen, and the ones that are treated are more susceptible. So there are lots of issues raised, but I think the key here is that, glyphosate doesn't affect bees, but it affects their microbiome. So whether that's an issue or not, we don't know. I agree that the paper doesn't answer that, but I don't think it's fundamentally flawed, right? Was the
glyphosate given at reasonable doses?
Yeah, it was at doses that they think they would encounter in nature. So, It's put on plants and then the bees would go eat it. Right?
So did they stumble onto something that they have to follow up on now? Is the microbiome of the bee the homing device that the bee uses? Because they maybe just got lost. Maybe they suffered from Alzheimer's and forgot where they lived. No, I'm serious. I mean,
this video, in the beginning, they have a guy saying that glyphosate causes everything, basically, and he rattles off 50 diseases.
What if the microbiome of the bee enables the bee to tell not only where the honey was, but also where the hive is?
I don't think that's unreasonable.
Right? And so you disrupt that and you screwed up the homing mechanism, and therefore you've gotten a result that says that they're all dead. Well, they're not dead. They're just flying around trying to find home.
So you're saying that the glyphosate makes it harder for the bees to round up?
Yes, in fact.
Well, anyway, Jake, take a listen to the TWiV, and tell us if you have specific issues here. We're happy to revisit it.
Oh, sure.
But it is a TWiV thing. It's not there's no viruses involved, as far as we know. Brianne, could you take the next one?
Sure. Izzy writes, "Hi, TWiVers. I've only recently started listening. Honestly, most of your episodes go a little over my head. I'm just a lowly public health BS." No, Izzy, that's not lowly.
No.
And it's not BS either.
Also true. So I usually have to spend a good amount of time pausing and Googling to figure out what you're talking about. I love doing that, though, because I encounter new things all the time. In a recent episode, TWiV 511, you interviewed Ned Landau. At one point, he mentioned ApoBac 3G, which, after much Googling, eventually led me to the field of restriction factors. From what I could understand, it seems that these restriction factors may evolve quite quickly, suitable for countering viruses that can mutate quite quickly. I was just struck by the dramatic nature of the whole scene, our cells and viruses duking it out on a relatively short time scale, each restriction factor and its virus a sparring partner. It seems to me such a moving example of evolution. I don't know if any of this makes sense, but would love to hear your thoughts on the evolutionary aspect of restriction factors. Thanks.
No,
she nailed it.
I just saw the movie in my head going.
That's it. You are not a lowly anything. You got it.
That's right.
You nailed it.
That's right. Yeah.
Yes.
And by the way, I spend a lot of time Googling to figure out what we're talking about, but I do it before each show. I have the advantage of
I was going to say the same thing. It takes me twice as long to read most books, because I'm Googling all the time. It's just a wonderful way to study stuff. Go for it.
That's right.
Yeah, I agree. And I also agree that restriction factors, particularly ApoBac 3G, are absolutely fascinating. And I think that it's a great example, and I'm glad you're as excited about it too.
So this is the prime example of arms conflict.
Yes, the Red Queen.
These genes have been in conflict with viral genes for many, many years, and people have begun to study this. This was first started by Michael Emmerman, Harmeet Malik, and then their trainees like Sarah Sawyer, Nels Elde, and many others. And we've done some episodes of podcasts looking at some of these papers. But, the idea is that these are among the restriction factor genes are among the most positively selected genes in the genome, because they have to change rapidly in response to viruses. And so we have definitely done many episodes on this. So, and you're right on, Izzy. It makes perfect sense.
Yep.
And they're super cool.
They are super cool. And there are many others besides ApoBac. Who's left?
I am.
Dickson, can you read Ricardo's?
I'll give it my best. Ricardo writes, I'll start again. Ricardo writes, Hello, dear friends of TWiV. Just sending 2 pictures of one of the talks of the 5th National Virology Meeting that is happening today in Port. The talk from Sao Jose Maria Andre about EVD68 and EV71 and acute flaccid myelitis. My best regards. Please extend my compliments to all the TWiV team for your wonderful dedication. Signed, Ricardo. And he sends a picture, and it shows a polio ward with everyone stuck in their iron lungs and nurses attending the patients, and it's a very grim sight.
There's another one.
And another one, sorry.
Can you translate the Portuguese?
No, but I'll be going to Portugal in December. I might come back and be able to do that.
After the eradication of polio, other enteroviruses like 68 and 71 have emerged as a principal cause of acute flaccid paralysis, especially in young children.
Exactly. And then he writes.
Yeah, go ahead.
And today I was in the 5th National Virology Meeting from the Portuguese Virology Society. Very good virology, full of food for our brain. Many TWiV fans, too. From virome to EVD68, CMV, HIV, non-permissive or non-susceptible cells, viral infection, etc. But I'm writing you because the last words of the meeting were from someone who was identified as one of the top virologists in Portugal, Professor Maria Odete Santos Ferreira. She died on the 8th of October of 2018. Her eureka moment was her involvement with the discovery of HIV-2 with Professor Luc Montagnier. Even at the age of 93, she was able to secure funds for the research of her faculty, pharmacy faculty, University of Lisbon, through grants. On top of her extensive dedication to science, she was also involved in our national program for the eradication of AIDS and was responsible for the program with the best results in the reduction of HIV-infected drug addicts. Say no to a second-hand syringe. The program allowed drug addicts to exchange syringes on pharmacies so they would not share contaminant needles. Taking this population from the most affected to the lowest one in a few years. There is, I'm reading, by the way, there is a small movie you can check out if you're interested, and she gives links to that. I know it's not of me to ask, but if you could share with the TWiV audience, I would appreciate. My best regards to you and, of course, to all the TWiV team, Ricardo. That's a great mail.
Cool.
Yeah, from Fernando Pessoa University in Portugal. You're going to Portugal, Dickson?
I am.
What are you going to do there?
We're going to have fun. We're going to eat and see and drink and be merry.
Those should be good things to do there.
I think so.
Speaking of fun,
let's
do some picks.
Okay.
Yeah.
Excellent.
Brianne, what do you have for us?
Well, I specifically wanted to pick something that was fun because I knew we were going to have that transition here. And I did search quite a bit in the archives to see if this had been chosen before. So this is a board game called Pandemic. Just to let you know, if you search Pandemic among the TWiV archives, you come up with quite a few things.
Yes. So
this is a board game where players play cooperatively against the game to try to combat 4 different infectious disease outbreaks that are striking around the globe. Each player has some different abilities, and you have to work together to be able to stop the outbreak. I've played this before with some of my friends. It's always a good time. And my students actually have an old copy they leave in the lab for times when they are doing incubations. And they every so often, my whole lab will be sitting around trying to get rid of these plastic square infectious diseases. So it's a lot of fun. There are expansions, though I haven't actually played them. And I thought people would want to know about a good way to spend some time thinking about infectious disease.
I like the subtitle. Can you save humanity? This was an electronic game also, right? Same thing.
Why should we bother?
There's a similar electronic game that I've played, but it's not exactly the same thing.
Okay. Cool.
I like the fact that it's cooperative. I think that's cool.
Yeah, I'd actually never played a cooperative game before this, and it's really fun.
Great pick.
Nice. Very good. Alan, what do you have?
I have something this is a technology and history thing that just really, really worked for me. The Shortwave Radio Audio Archive. So what these folks are doing is they're are radio stations, obviously, around the dial that broadcast, and they are collecting recordings of, in many cases, individual stations, but also the ones that are really cool are these recordings where they record an entire chunk of the radio spectrum. For example, the entire AM dial. And what you can then do with it is go back to that, and when you're playing it back, you can actually tune around between stations as if it's in real time. And it turns out the technology to do this, at least if you were really, really dedicated to it, was available as early as the late 1970s. So people started recording these chunks of radio spectrum onto videotape because it had enough bandwidth that they could do this. And this group is now digitizing those and making them available online. If you don't want to dig into all that, do at least watch the video, because he gives a little explanation of what's going on and then tunes around the radio dial from an evening in 1986. And it is a freaking time machine. There was a point about 3 minutes in where my jaw just hit the floor, and it was amazing. So give that a try. Amazing. Rich Condit, what do you have?
So last time I was on, which was 3 weeks ago, I picked James Cook's journal of his first Pacific voyage, which I was partway through at the time, and I just finished it last night. And I said that I was reading it with my, this is a shout out to Izzy again, with my iPhone in one hand, using Google Maps to figure out where he was, and always occasionally looking to see if I could find a better map on the internet. And almost too late, I stumbled into this thing, which is an interactive map of his voyage. So this shows the route for his whole voyage, and you can, starting off with a course resolution, you can click on submaps that show a portion of the route. And as you click on that, it gets deeper and deeper into more detail in places where he lingered around a certain coastline for a while. Mm-hmm. And it's got every single date. And if you click on the date, his journal entry is there.
Wow.
So somebody has done the whole thing, and it's amazing.
That is
really cool.
Wow. And
I recommend, once again, the journal, because it's just fascinating reading, for me anyway. Mm-hmm. And I mean, this is you're looking at exploration, through the explorer's eyes. And he was one smart dude, and he did a very, very thorough job. He was really a good scientist. So the journal is terrific, and this is a great supplement to that.
Very
cool.
When I first saw it, I thought you were doing last week's pick again.
No.
I saw James Cook. I said,
this is an embellishment on it.
What is he doing?
Right.
You got something about James Cook, huh?
Yeah.
You like those explorers.
Yeah. I'd like to read to start. I was talking to my wife today. It'd be nice to find some sort of a volume that had that whole era of European exploration outside of Europe as a sort of a summary. I'd have to probably go back and get my grammar school history book or something like that, but something a little more sophisticated than that, and then dig down deeper, because these were amazing expeditions.
They were.
Yep.
Yeah, I got into the Cook voyage. I read several books about it several years ago, and just an amazing project.
Yeah.
It's amazing how brave they were too, because
he was gone for 2.5 years.
They had no idea what was going to happen.
He went aground on a reef inside the Barrier Reef in Australia, thousands of miles from anywhere,
right?
And just barely managed to salvage the ship and limp into Batavia to get the real repairs, where, of course, then he encountered dysentery.
Right.
And on and on. It's quite a trial.
These explorers of yore
very brave.
used to be gone long times.
They were.
Yeah.
Well, and when he ran aground in Australia, not only was he aground thousands of miles from home, he was aground in a continent he had just discovered.
Right.
Yeah.
Right.
Which I can't even imagine what that would be like.
Exactly.
Nobody knows where you are because you just found it.
Yeah. I had the pleasure of standing at the base of his mountain in New Zealand for about 3 hours with a camera focused on the mountain, and I was taking time lapse, and I was just, the sun was coming and going, and I can put this whole thing together now like a flip, like the comics when you flip it, and you can actually watch the mountain light up it was fantastic. So
I not too long ago finished the what is it? The Plight of the Dodo? The Song of the Dodo.
Song of the Dodo.
Oh, yeah.
Right.
And they describe Alfred Wallace, who came up with the theory of evolution about the same time Darwin did, but didn't get any credit for it.
Oh, he did now.
He did.
Yeah, but at the time they tried. At the time they really, I mean, the powers that be were pushing Darwin. Anyway, he did a lot of traveling in the Pacific, and he would get to islands. He's by himself. He would get completely bitten by bugs so much he couldn't even collect. His feet were a mess, and he would have to leave, and then the rains would start, and everything would get ruined. He once had a huge collection of things he'd collected back, and they were gone.
Yeah.
Oh my gosh.
Yeah. It was a tough life.
Dixon, what do you have?
I have, instead of giving my pick of the week, I gave about 2,000 picks of the week through an Instagram site on the Scientist magazine. Yep. And if you subscribe to the magazine, you can send in a picture of your choice, and it's usually high-def picture of a wonderful scene of some sort, unlabeled. So you can try to guess what they are.
Oh, and then they post them?
Yeah.
Dixon, do you know what Instagram is?
I do now.
What is it?
It's a way of posting pictures up to let you know that you're aware of something that everybody else can share with you.
They show you periodically pictures, and you go, "Where'd you get that picture?"
Yeah, I know.
Dixon, it's Instagram.
I got it.
But my picture, the pictures on the Scientist are not showing up for me.
Why?
Are they showing up for everyone else?
I got them.
It must be our Columbia internet.
Of course, it's our we've been blocked.
Yeah, I don't see any of the pictures.
But they're not labeled. The thing is that they're not labeled.
Well,
so
I don't care, but I don't see them. Do you see them on your thing?
If you click on them, you get the full caption.
Yeah, but
you get
it's better to just scroll through it and see how many of these things you can guess.
Subscribe. Do you know I have an Instagram account, Dixon?
I don't know that.
Do you use Instagram? Of
course not.
Why not? Don't you want to see pictures?
I'm not a social, but no, I do. And in fact, I that's why I put this one up.
Okay.
Dixon, sometimes Vincent puts pictures of you on his Instagram.
I know he does, and I get furious because some of them I don't like at all, but you put them up anyway.
Whoa.
We'll discuss this later.
You've never seen any of them.
I've seen all of them because you type my name out on Google search and you get them all up.
Is that right?
Yeah.
I have one of you sleeping. That's the only bad one.
No. There was one with my eyes spread wide open. There's something in the background.
It's cute, though. It's very nice.
It's not. No, it's not.
It's very nice. Don't worry, Dick.
Not if you're me.
When did you start serving your guests these delicious-looking sandwiches?
Exactly.
Oh, that was just a one-off thing.
Oh,
aren't they nice looking?
Yeah, I didn't get that.
We had a guest, and we decided it would be quicker to have him here with a collaborator and give him lunch rather than go out. And I thought of captioning it, "If only sandwiches could speak." What would they say?
They would say, "Ouch." Please don't eat.
I have over 800 photos on my Instagram.
It's amazing.
All right, my pick is an amazing site put together by the Royal Botanical Gardens, the Kew Royal Botanical Gardens. It's called State of the World's Fungi 2018. It's everything you wanted to know about fungi. Not just what kind of mushrooms, but diversity, the tree of life, new discoveries, useful fungi. Oh my gosh, the things, food, medicines, biofuel, plastics, vitamins, research, agriculture, washing powders. Did you know that Lego plastics are made with a enzyme from fungi?
Did not.
I did not.
No.
All the things that fungi are used for, it's all summarized here.
What's it called? Snap Ace?
Not Snap Ace. And they have wonderful graphics, photos, information. It's just amazing. They did a great job.
Do they cover mycorrhiza?
Yes, of course.
Good.
Yeah. This is cool.
They cover things that you just...
And all the fermentation products.
Oh, everything. I mean, you click a category of what bioremediation.
Look at that.
They can degrade a variety of pollutants.
You bet.
Pesticides, explosives, dyes.
Yeah.
And they give you the names of them. It's really cool. So check it out.
Indeed.
This will be great for my microbiology class on this
For sure. It's really good. And we have a listener pick from David Spector, who I saw last week at Penn State Hershey. David is a fan, and he writes often, and I went to dinner with him, and he came to both of my seminars and to TWiV. Thank you, David. And he writes, Dear TWiVniks, thanks to Vincent for his visit to Hershey last week. Here's a listener pick. It's a story about Bruno Latour, a French philosopher, and his lifelong love affair with science. He argues that science strives to attain an increasingly more accurate description of a reality that lurks beyond our senses and their technological extensions. The process of doing so is very messy, nonlinear, and all too human. Fair enough, and something with which probably most or all of us would agree. More controversially, he says that facts don't speak for themselves. Rather, their authority derives from the culture and society in which they are generated. Seems like the evidence for this position has strengthened considerably in the last 2 years or so. There are implications for how science communicators should approach the challenge of science denial. The latter arises out of a... Not arouses. The latter... Sorry.
Now you're reading like I am.
The latter arises out of a subculture that maybe was growing all along in parallel with the growth of the scientific enterprise, and more recently has been juiced by post-truth positions not nearly as nuanced as Latour's. For these deniers, claims of the authority of science have no value. I suspect that for the zealots, once they have joined that community, they are a lost cause. We probably shouldn't waste our time with them.
What's Latour's dates? When did he live?
I'm looking, Dickson.
Okay.
Pulling up the article, which is a New York Times article.
Right.
He lived recently because just in 1996, he was approached by a developmental psychiatrist.
Okay.
So he's I don't know if he's still alive, but
could find that out too.
You could. Let's see. Better like Twix to build a wall of informed non-scientists.
And build a wall.
Exploring the exhilarating and messy process and demonstrating the inherent and utilitarian value of the knowledge we gain. That's a great way to put it. I really like that. Keep on twivin', Dave. David's a professor emeritus in microbiology and immunology at Penn State Hershey, which is where I visited last week.
That was a good show.
Hello to everyone there. Thank you, David. Thanks to everyone who hosted me. It was a fun
It's the only city in the country that you can visit and get a kiss without your wife getting jealous.
All the streetlights are Hershey's Kisses. And we had dinner at the Hershey Hotel. It's a very interesting town, by the way. It was built by Hershey, planned town to house his company.
Absolutely.
Surrounded by the farms that provided the milk for his chocolate. Yeah. And then when the Depression arrived, he built the Hershey Hotel to keep people employed.
Interesting.
It's a remarkable, Depression-era hotel.
Yeah.
Really remarkable. And they had to show me this. They said, let's go up to the check-in desk. And he told the guy, he said, do your thing. And the guy took out 2 Hershey bars. He said, milk or dark? When you check in, that's what you get.
Right. One for each pillow.
Very cute. Yeah, they dominate the town. It's and listen to this. Last thing. When Hershey died, he had no children. He had founded a school in town for disadvantaged children. And they live there. It still exists. He gave all his money to this school. $8 billion.
Whoa.
Wow.
And he died quite some time ago.
Wow.
So this little school of 1,200 kids and their teachers has a ton of money. It's amazing.
Wow.
Of course, he also paid for half of the medical center. That's why it's in Hershey. Because this is part of Penn State over on the other side, right?
That's true.
And they wanted to build a medical school. And he said, build it here and I'll give you $50 million.
That'll do it. And
that'll do it for TWiV 519. You must listen on a phone or tablet. You use an app. Please subscribe to TWiV. Dickson's tablet's not working, but subscribe so you can get every episode and we get the numbers. If you really like what we do, go over to microbe.tv/contribute. You can give us $1 a month if you'd like. We'd appreciate it. And questions and comments to TWiV at microbe.tv. Dickson, Despommier, Racaniello.org, thelivingriver.com. Thank you, Dickson.
You're welcome. This was, as Kathy would say, this was a lot of fun. She would.
She would?
She would.
Yeah, that's what she'd say.
She'd probably have.
That's her line.
She's probably having more fun watching Hamilton. Brianne Barker is at Drew University over in New Jersey. She's on Twitter as BioProf Barker. Thanks, Brianne.
Thanks. It was great to be here.
Rich Condit is an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
And Alan Dove can be found on Twitter as Alan Dove, and he's on turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. I'm at virology.ws. Thanks to ASM for their support and Ronald Jenkins for his music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.